Hello everybody, I am Dinesh Urpi. In this video, we'll continue with the study of gene technology, immunology and computational biology paper. And in this video, we'll study the gene cloning vector concept of plasmids and one plasmid, that is PBR322. So we'll study in brief. So what is a vector? What is a gene cloning vector? So in recombinant DNA technology, what we do is we isolate the gene of interest from one organism and then we transfer it to the target cell or target organism. So to do this, to transfer the gene which is isolated from one organism to the target cell, we need a transport, we need a vehicle which can carry this DNA. So that job, that function is done by vectors. So through the vectors, using the vectors, the gene of our interest can be transported. So for the formation of recombinant DNA, a vector is used. So vector is a DNA molecule, often plasmid or virus. So that is used as a vehicle to carry a particular DNA segment into a host cell as part of a cloning or recombinant DNA technique. The vector typically assists in replicating or expressing the inserted DNA sequence inside the cell. So there are different types of vectors which are used. Here you can see the plasmid vectors. So plasmids which are isolated from the bacteria used as vectors, and phage bacteriophages. So they are viruses. So DNA molecule which is extracted from viruses, they are also used as vectors. And cosmid vectors which are synthesized using different techniques. So they are also used as vectors in the recombinant DNA technology as gene cloning vectors. A plasmid is a small extra chromosomal DNA molecule within a cell that is physically separated from chromosomal DNA and can replicate independently. They are naturally found in bacteria. So plasmids were used as vectors in the beginning. So these plasmids are nothing but the extra chromosomal DNA molecule. So here you can see this is a bacteria, a prokaryote, and inside this bacteria it has its own chromosome, own DNA or genetic material. So this is the genetic material of the bacteria, which consists of all the genes necessary for the functioning of this bacteria. Now along with this main genetic material, in the cytoplasm of the bacteria, we find small circular double-stranded DNA molecules. So different types of small circular double-stranded DNA molecules are found. So these are the plasmids. Different species of bacteria have different number of plasmids. So different types of plasmids. So many of them are isolated and used as vectors in recombinant DNA technology. So gene cloning vectors. So properties of an ideal vector. So to use a plasmid or to use a phage as a vector, it must have some properties, some characters. So what are those? It should be small in size. So smaller the DNA molecule, it will be very easy to manipulate. So that is the main idea. It must have an origin of replication. So origin of replication is a site from where the DNA replication starts. So if this is a vector, we are going to insert the gene of our interest into this, and this will be introduced into the target cell. And once it enters into the target cell, it has to multiply. So if it is a cloning vector, it is going to multiply. So for multiplication, there is a need of origin of replication. So origin of replication is the point from where the DNA duplication or DNA replication starts. So it is a must in a vector. It must also be compatible with the host organism. So the vector which is going to be introduced into the host into a target cell, so it must be compatible. If it is not compatible, it will be destroyed by the host cell machinery, so it will be of no use. So this is another character. It must possess a restriction site. So this is very important, because restriction sites, we already understood, they are the sites in which we are going to introduce the foreign gene, the gene of our interest. So restriction site is the one where the restriction endonuclease enzyme are going to act and they are going to cut the DNA. So to introduce the gene, you have to cut it. So for that, restriction sites are needed. Without restriction site, a vector cannot be used. So that is another important property which must be present in a gene vector. The introduction of donor fragment must not intervene with the self-replicating property of cloning vector. So the site of introduction of gene must be such that once the gene of our interest is introduced, once it becomes a recombinant DNA, it must not affect the normal replication process of this vector. So the gene should not interfere with any other genes which are present in the plasmid or the vector. And a selectable marker. This is another character which is expected from a vector. Now, once we introduce the recombinant DNA into the host cell, into the target cell, we want to screen it. We want to know whether it really got incorporated or not. So for that, a selectable marker must be present. Normally, the selectable markers are present in the form of antibiotic resistance genes like ampicillin resistant gene, tetracycline resistant gene, etc. Using that, we can screen, we can identify the cells with recombinant DNA and the cells without recombinant DNA. So that is the selectable marker. So selectable markers help in screening of the recombinant DNA containing cells. It should be capable of working under the prokaryotic as well as eukaryotic system. So because we are going to transfer, we are going to introduce these gene vectors into prokaryotic cells like bacteria or plant cells, animal cells, etc. So therefore, it must be capable of working under both prokaryotic and eukaryotic system. Multiple cloning sites must be present. So more cloning sites, so restriction sites. More the restriction site, it will be of more use. To introduce, you can introduce more genes with the help of more restriction sites. Types of cloning vectors. Cloning vectors are basically classified into two types: cloning vectors and expression vectors. So commonly we use the term cloning vectors, but vectors which multiply, which become multiple copies once they are introduced into the target cell, they are commonly called cloning vectors. Whereas the vectors which express the genes which are introduced in the form of protein, enzymes, etc., they are called expression vectors. So here we can see here a cloning vector is a small piece of DNA which is used to introduce the foreign gene of interest into the host cell. It can be stably maintained within the host organism. So cloning vector, uh, as we already studied, it is a small piece of DNA. It is used to obtain multiple copies of the foreign gene of interest. So this is the main purpose of the cloning vector. So here, when we prepare the recombinant DNA, let us say we have prepared only few number of recombinant DNA copies. So only one recombinant DNA copy, it enters into the bacterial cell or some other prokaryotic or eukaryotic cell. It will multiply because plasmid is a self-replicating DNA, and when it replicates, the gene which is introduced into that is also going to replicate. So like this, we are going to get multiple copies of the gene. So in the modern gene technology, we use PCR machine, so polymerase chain reaction machine. Earlier, this technique was used to obtain multiple copies of the gene. They can be plasmids, cosmids, and phages. So cloning vectors, so basically they are plasmids and cosmids and phages, and BACs, YACs, and MACs. So here I have given bacterial artificial chromosome, yeast artificial chromosome, or mammalian artificial chromosome. So there are different types of vectors, maximum number of vectors which are cloning vectors. A typical cloning vector consists of an origin of replication, unique restriction sites, reporter gene, and an antibiotic resistance site. So these are the minimum requirements which are present in the cloning vector. So let us come to the expression vector. Expression vector is a plasmid that not only introduces a gene of interest into host organism, but also helps in analysis of the gene of interest via relevant protein product expression. So this is the main difference between cloning and expression vector. Here the gene which we have transferred, it is going to copy itself. So we are going to get multiple copies of the gene. But here, the gene which we have inserted, we are going to obtain the product of that gene. So which type of enzyme, protein that is going to produce, so that will be of interest here. So we use expression vectors wherever we need the gene product, right? So it is used to obtain or analyze the gene product, which may be RNA or protein of the inserted gene of interest. Only a plasmid can be an expression vector. So when it comes to the cloning vector, we have the choice of plasmids, cosmids, and phages, BACs, YACs, MACs, whereas for expression vector, we can use only the plasmid. And features. An expression vector consists of regulatory elements like enhancers, promoters, termination sequence, transcription initiation site, translation initiation site, in addition to having all the features of a typical cloning vector. So because here the gene is necessary to produce its product, not only the gene, along with the gene, we have to insert the cassette. We call it as a cassette. That means the genes, the sequences which are necessary for the expression of that particular gene. So try to understand the difference. In a cloning vector, we are going to introduce only the gene. So that gene gets multiplied. So if our purpose is to get the product in the form of RNA or the protein, so we need to insert the gene because that is the one which we are going to study. And that gene to express, we have to introduce all the sequences which are necessary for the gene expression, like regulatory elements, promoters, operators, initiation sites, termination sites, etc. So that is how the expression vector is used, and mainly to study the product of the gene. Now let us study one plasmid vector which was very popular, PBR322 PUC. So like this, there were many plasmids which were used in the earlier stages. So PBR322 plasmid vector was created in 1977 by Bolivar and Rodriguez. So that's why it is called PBR. P means plasmid, B refers to Bolivar, and R refers to the Rodriguez because they have created it. So that name is given PBR322. First widely used cloning vector in molecular biology. So this was the one which was prepared in the beginning, and it was very popular in the early days of biotechnology. It is a circular double-stranded cloning vector, 4.4 kb length, so 4400 or base pair length. So that is the size of the DNA molecule. It is double-stranded and it is circular. So it has, as we already studied in the previous slide, all the properties are there, like origin of replication site, restriction sites, then antibiotic resistant genes. Okay. So like that, the basic necessity of a vector are all present here. Right? And this PBR322 actually had around 40 restriction sites, about 40 different type of restriction sites are present in this PBR322. So PBR322 structure has origin of replication, restriction enzyme sites, and selectable marker sites. So we already discussed about it. Origin of replication for multiplication or replication, and restriction enzyme for the introduction of gene of our interest, and selectable marker, that is nothing but antibiotic resistant genes for the screening or selection procedure. The origin of replication in plasmid PBR322 is known as PMB. That is the naming of the site of origin of replication. The copy number of this plasmid is 15 to 20. So whenever we talk about plasmids, we use a term called copy number. Now what is copy number? Copy number is the number of molecules that can be found in a single bacterial cell. So higher copy number and lower copy number. So in many cases, the higher copy number is desired, and in many cases, lower copy number is desired. So the copy number of PBR322 was very useful for using it as a vector, gene vector. Restriction enzyme sites. So to continue with the study of PBR322, PBR322 has around 40 different restriction sites. So it's a very high number of restriction sites, and that condition makes it a very useful vector. Almost 11 different restriction sites are present in the region of tetracycline resistance gene. Now, the restriction sites which are located within the antibiotic resistant genes are very important. Just imagine. So there is, let us say, this is the ampicillin resistant gene, ampicillin resistant part, and this is tetracycline resistant gene. So from here up to here, this is tetracycline resistant gene. So the presence of this gene gives resistance against tetracycline. That means the bacteria cannot be killed by the tetracycline antibiotic, because the gene product of this particular gene will give the resistance. Now there are restriction sites. So there are around 11 restriction sites which are located here. Just imagine, so there is a BAMH1 restriction site here. If I use BAMH1 endonuclease enzyme and I introduce a gene here, so what happens? I am going to break the tetracycline gene. So in between the tetracycline resistant gene, I am going to introduce the gene of my interest. So this particular plasmid is going to lose the tetracycline resistance. So this property becomes very helpful in selecting the recombinant DNA, identifying the cells with recombinant DNA. So just imagine the bacteria which is having the resistance against tetracycline means there is no recombination. A bacteria which has lost the resistance. Why it lost the resistance? Because I have introduced the gene here using this enzyme. So if the gene is introduced, a foreign gene is introduced here, it loses. So based on that, you can screen. So that is the importance of the presence of antibiotic resistant sites. So some of the ampicillin resistant region also has 9 restriction sites. Some of the known restriction enzymes are BAMH1, HIND3, EcoR1, SA2, etc. Selectable marker sites. 2 selectable marker sites or antibiotic resistant genes are present on the genome of this plasmid. So I already explained it. This is one selectable marker site and this is another selectable marker site. Antibiotic resistant sites are called selectable marker sites. So the ampicillin genes code for beta lactamase, which can be used for screening microorganisms when a foreign DNA is being inserted in the plasmid. So this I already explained how the selectable markers are of use during the screening. What is the advantage of using pBR322 as a gene cloning vector? Due to its manageable size, pBR322 is widely used as a cloning vector. The presence of two antibiotic resistant genes eases the selection process of recombinants. Multiple restriction enzyme sites make the plasmid compatible in many ways. It has a high copy number, which is highly favorable in genetic engineering. So the presence of two selectable markers, presence of high number of restriction sites, and small size of the DNA molecule, and copy number. So all these characters make PBR322 as a very useful vector, gene cloning vector, and it was widely used in the recombinant DNA technology. So that's about the structure and some characteristic features of one very famous plasmid vector, that is PBR322. So that's for this particular video. And again, I'm mentioning the references which I have used, and I recommend the students to go and search the textbooks or the internet for more information. So thank you.